And we expect to have some time for questions at the end as well. Thanks very much. And our next speaker is Dr. Manrai from Harvard.
Hi everyone, my name is Arjun Manrai. I'm a researcher at the Department of Biomedical Informatics at Harvard Medical School. Delighted to be speaking today about reproducibility in the context of biomedical computing and large databases. Many of the examples will come from human genetics. That's what we spend a lot of time working on, as well as the CDC and Haynes data set, but I think many of the lessons apply to the topics that we've been discussing today. First, I just wanted to thank the organizers for inviting me. So I don't think I need to motivate reproducibility to this crowd, but I thought this was a good way to start. This is the great 20th century philosopher of science, Karl Popper, who said very directly that non-reproducible single occurrences are of no significance to science. So reproducibility is, of course, very old as a concept, but it's been all over the news lately. It's been very prominent in drug development. We've heard some of that from the previous speakers. But really, in biomedical computing, this affects the full translational spectrum from basic science to clinical trials. There's even quite a popular blog called Retraction Watch that follows retractions in science. And there's been, quite a bit of debate over numbers like the 6 of 53 from the Amgen study that looked at previous landmark cancer papers. And this has led, to wonder about the reproducibility of these reproducibility studies themselves. This, of course, may actually strengthen the conclusions of the original studies while potentially casting doubt on their methods. But either way, I think there's widespread consensus, as we can see in this meeting, that this is an important topic to address and to come to terms with. There's been many solutions, as you've heard, to help bolster or improve the reproducibility in biomedicine and animal science. I think the top thing that I see is individual responsibility. So this is better training in statistics. This was a study that was published in Nature. It was a survey of 1,500 scientists last year, and you can see that the top two factors are better understanding of statistics, better mentoring, supervision. Many of those other factors, I think, are individual investigator responsibility. So the question we have generally is, what about structural factors? What if there was no bias? What if there was perfect essential investigation itself by the investigator? What factors outside of the individual investigator's control would shape the reproducibility of their conclusions? And so where I think this is really fundamentally important is in the context of multiplicity. And so this was the initial observation that prompted the results that I'm going to discuss. These are two scenarios, and the one on the left is just a SNP chip. It might have a couple 100,000 SNPs on it that we measure in a GWAS or something like that. And we almost always address multiplicity explicitly and rigorously in that context. And if we have a large shared database, which I think is very common now, we've seen many examples of that today. In human genetics, it's also very common. There might be, in a process that spans decades, that we see many investigators analyzing this shared database together, and we almost never explicitly address multiplicity in that context. But of course, this process on the right here can resemble high-throughput genomics in scale, but not in synchrony. And if we're not very careful about how we address multiplicity in this context, we can end up in very poor reproducibility situations. So how can we study this problem? So when I was in grad school, I saw this paper. It was very inspirational. I'm sure many of you have read it as well. It's called Why Most Published Research Findings Are False by Johnny Anides. And you can see that it's been extremely influential. There's over 2 million views on this PLOS Medicine article. I'm responsible for about 1 million of those. What it does is it gives us a framework and a language and a way to think about the reproducibility of a given scientific field in terms of this concept of PPV, which is very familiar from the diagnostic testing world, the positive predictive value of science, not of a given test, as a function of what is happening in the scientific community. And so the key parameters here, I've just highlighted two. There are many others in John's framework. R, which is the pre-study odds. It's the ratio of non-null to null relationships that are being studied in the field. And U, which is a bias term, which reflects the proportion of findings that are published that wouldn't be published if studies had been conducted perfectly to begin with. So our goal was to extend John's framework, and so this is work with John. I was very fortunate to connect with him in grad school. The goal is to extend this framework. So many of these parameters in his original framework were scalars or constants to these communal science situations in which many investigators are conducting these studies with studies of varying quality. And so in the setting of communal analysis of a shared data set, we have to generalize this model in order to parameterize reproducibility. So there's N and B. These are two big terms. I'm not going to dwell on the math. I'm just going to give some intuition here. There might be relationships being studied by only a small set of teams, or there might be certain relationships that have 100 teams studying each of those relationships in the given data set. And then, of course, as we've seen also, each study in this given analysis might have very different powers. So there might be some studies with 80% power, very rigorously enforced, and others with 10%, 20%, 30% power. And so then the last part of this is recognition and sort of explicit accounting for the fact that this quantity that we're defining, dPPV, the data set positive predictive value, is a stochastic process. It's a random variable itself, and it has variance even in these any of these settings that we're looking at. So the distribution that's sort of central to this quantity it's indicated by PB there. It's a Poisson binomial distribution. It's the generalization of the binomial distribution for non-equal probability of success on each trial. And this is a ratio distribution. It's hard to manipulate. We can do Taylor approximations and compute the expectation and the variance of this quantity and do a lot of analysis this way. So a lot of our work has gone into making this quantity analytically tractable. But where I think it's more interesting is thinking about it in the simulation setup that we've been working on. And so these communal science settings are ubiquitous. There are a few examples shown here. The primary example I'll talk about today is NHANES. This is a CDC data set, National Health and Nutrition Examination Survey, but we've seen very similar results with large-scale genomics data sets, for example, the Genome Aggregation Database and the Mouse Phenome Database as well. And so I've been discussing theory, but briefly to motivate this a little bit more, this is all in service of medicine. In the context of human genetics, we published a paper last year where we found, to our surprise, that multiple variants had been misclassified, and these variants were much more common in African Americans. So when we looked at patient records at a leading testing lab where individuals had misclassifications, they were much more disproportional in African Americans than other populations. So this type of irreproducibility is what we're trying to avoid with these frameworks. So this is our computational framework for studying communal science and measuring this quantity, dPPV, the data set positive predictive value. We start with a shared data set, and so this is NHANES for us typically in these analyses. And then essentially we use empirical distributions of that data set to simulate artificial variables that follow the same distributions so that we can look at pairwise correlations and control the fraction that are considered null or non-null over time. And what we do is we simulate different teams gaining access to this data set iteratively in time steps, performing studies with different type 1 and type 2 errors. For example, in the context of NHANES, this is looking at the relationship between albumin and total calcium. Bias is used to select which findings are published, and then because we seeded this process, and this is all in a simulation framework for this data set, we can measure this quantity, the data set positive predictive value, over time. So this is the simplest possible scenario here, but I think it really illustrates the central point about multiplicity. So suppose there are 1,000 relationships that are being studied in a given shared data set. There's one study being conducted per relationship, and a relationship is, for example, the correlation between albumin and creatinine, or albumin and calcium, whatever we're interested in. There's the customary significance threshold of 0.05, and the pre-study odds, which is the ratio of non-null to null findings, is 0.001. And we have good power. Nominally it might seem that we're well set up here to do rigorous science, but if only the studies that survive that 0.05 threshold are reported, in expectation, the reproducibility of the claimed findings from this data set is less than 2%. And so the reason is that the power of our study, which is 0.8 or 80%, exceeds the false positive rate by a factor of 16, but the null relationships dwarf the non-null relationships by a factor of 1,000. So we're essentially doomed here. So that's the simplest possible scenario. The way a data set is analyzed in practice, I think, is well captured in this article by Gelman and Loken in 2013, where they invoke the Borges story, the Garden of Forking Paths. So essentially, you can chop up a variable, you can binarize it however you want, you can adaptively set your analysis after you see the data, and this is not necessarily p-hacking. It could be p-hacking, which many of you, I'm sure, are familiar with, but this could be, I think, very innocent sort of post-data inspection corroboration of your hunch hypothesis, which I think, I guess, broadly would be confirmation bias. But really, the way the data set can be analyzed can be very, very complicated. And this article does a good job of giving practical examples of that. So in the context of Van Haynes and looking at the shared data set, what hurts reproducibility is when teams arriving late, for example, tend to essentially pursue a priori less likely hypotheses. And so the key parameter here is R, which is the pre-study odds, the ratio of non-null to null hypotheses. It's shown in red here on the figure. And essentially, if this declines over time, then the DPPV, or the dataset positive predictive value, will as well. And so, for example, most a priori likely relationships might have been claimed by the early investigators who first had access to the dataset. And as the dataset was opened up to the broader community to essentially pursue novel hypotheses, what you'll oftentimes see is that a priori less likely hypotheses are pursued. And this is in the background of how this dataset is being used, but this quantity will fall over time. So what helps on a more positive note is if we are able to compensate for pursuing less likely hypotheses, of course, with greater power. I think a good example from human genetics is going from the candidate gene era to the GWAS era, where the first GWASs, the genome-wide association studies that were conducted, we learned about the effect size spectrum, and then we increased sample size considerably to the tens of thousands and even hundreds of thousands to find significant associations. And I think what you've seen there is lower effect sizes than initially hoped for, but tremendously reliable results with respect to what was previously done in human genetics. There's a lot of counterintuitive findings that we see. So this is looking at the studies per relationship. So the blue bars on the top are the fewest studies per relationship, all other things being equal, and the green bars are in the middle, and the red bars are the most studies per relationship. And so this was pretty counterintuitive to me at first. If there are many studies essentially being conducted by independent labs for a given relationship, we would normally think that type of corroboration is helpful for reproducibility, but what often happens in practice is that they're selectively reported, and the ones that are reported from that set tend to be less reproducible over time in our analyses. So those are summary patterns of the expectation of this quantity that we've developed, but of course there's variance around those values as well, and so each of these dots is a different realization of this stochastic process. And what we've looked at here is how liberal the data governance is, and what we mean by essentially as a proxy for that is how many studies are being conducted per data set. And so in the green here is very liberal data access policies where everyone can conduct one of these studies, but they're not overstudying a given relationship. And on the red is very restrictive data access policies. And as you might expect, this quantity itself is much more variable when we have very restrictive access policies. because essentially the quantity scales with the inverse of the number of studies being conducted. And so I think counterintuitively to me, what this means is if a reproducibility audit of a shared data set is being conducted, it might make sense to conduct it on a small set of studies, but that might be the most noisy and the least reliable essentially setting to conduct that analysis. So I think, when you're going through the literature, it can feel like this. I think if you remember one thing from the talk is that these structural factors and structural context can help tremendously in understanding reproducibility. I think just in summary, shared data sets, they facilitate rapid, simultaneous testing by many teams. I think this is a multiplicity challenge that we haven't really addressed that really rivals high throughput genomics, but is handled completely differently. The multiplicity in these settings should be addressed. I think we've pitched essentially just one way to do that. There are many others. Reproducibility in a shared data set, I think, should be viewed as a stochastic process, and its variance should be appreciated. And then structural factors, really, we found can be as influential on reproducibility as individual investigator responsibility. And last, that these reproducibility audits depend on variance. So I just wanted to thank my PhD supervisor, Zach Kohani, our co-authors here, and colleagues Johnny Anidis and Chirag Patel, Zach's lab, and then the organizers for inviting me. Thank you.
Thanks very much. And our next speaker, final speaker for today's session, and for today is Dr. Jonathan Kimmelman from McGill. Thank you.
Great, thanks very much. My focus is going to be primarily on cancer. My talk's going to divide into 4 parts. And I want to start by talking about what I think of as the paradox of personalized or precision medicine and why this may be creating more pressure and demand and burden, in a sense, on the way we do preclinical research. So the project of personalized medicine or precision medicine, whatever you want to call it, is primarily aimed at taking heterogeneous populations and breaking them up in, that have highly variable response to drug, and to breaking them up into various strata that have more homogeneous responses to drug. And this is a wonderful project. It's a project that has returned many dividends, particularly in the realm of cancer. But it's a project that in some way I want to suggest is chasing its tail, and that may again be creating a very heavy demand for and expectation about the way we use preclinical evidence. And I want to suggest that there are 5 ways that there's a paradox that's sort of built in to the mission of precision medicine. The first is a really obvious one, which is that the minute we take large heterogeneous populations and divide them into various strata, we are stuck with smaller populations, which by definition, because they have lower statistical power, we're dealing with a higher degree of variance in terms of characterizing the properties of a drug in that population. And that means that if we're dealing with really small, finer grained strata of populations, we may not necessarily be able to rely on the standard model of the randomized control trial to make clinical inferences. Ultimately, we may be needing to combine different forms of evidence. The second major challenge or paradox that's built into precision medicine is the notion of edges and boundaries. The goal, of course, is to develop diagnostic techniques that we can allow to stratify patients, but anytime you have a diagnostic technique, typically you're dealing with some kind of a cut point on that diagnostic, and of course, as we know with genes, you might have a particular variation of alleles, you might characterize with a high degree of clarity or resolution the response of an individual to a drug with a given allele, but you may have a lot of uncertainty about these various other alleles. And so the number of different diagnostics you use, the layers of diagnostics you're putting on top, are always raising questions about the cut points and about the generalizability of that precision medicine technique to other similar but not necessarily identical patient populations. So again, we have this proliferation of imprecision in some ways by virtue of the fact that we have boundaries or edges with the ways that we classify patients into finer and finer grained strata. The third issue is one that was talked about a little bit, I think, in the second session, and it's the issue of algorithms and interpretation of data. There is, to my knowledge at least, only one published randomized controlled trial testing algorithms to match patients to treatments in the context of cancer. It was the Shiva trial, or I think it's supposed to be pronounced Shiva trial. It was unfortunately a negative trial, but the interesting thing about this trial is the question of what kinds of information and inputs are used to inform the algorithms that we use to match drugs to patients in the context of cancer or elsewhere. This is a publication by a group in Canada that has explicitly decided to reject any kind of preclinical evidence in their patient matching algorithms because of concerns about reproducibility. But that's hardly the consensus view. If we look to, for example, a consensus statement issued by ASCO and a few other professional societies, they classify preclinical evidence as grade D, but still clinically actionable in the absence of other higher forms of evidence. So here there's clear endorsement of use of preclinical and animal studies to inform patient treatment. Here's another example. This is a study that used whole exome sequencing to identify clinically actionable sequences in patients with cancer. And you see that the most frequent category of clinically actionable mutations are informed by level D evidence. This is preclinical evidence. Okay, so that's another way in which there's precision medicine maybe sort of creating greater demands for preclinical evidence. The fourth issue is the issue about the rapid evolution of our knowledge and the techniques. By techniques, I mean the techniques that we use to diagnose patients. The diagnostic techniques themselves may be undergoing evolution, but also the algorithms we use may be undergoing evolution by virtue of the fact that we're always accruing more information about the properties of various drugs or treatment approaches. So going back to the Shiva, it's a wonderful study, a negative result, unfortunately, but by the time that this study had been completed, probably the algorithm for matching patients to the drugs is already outdated. So you never really have any stable ground to stand on in virtue of the fact that the algorithms that are being validated in trials are undergoing evolution while they're actually being tested. And the fifth issue is one that is kind of pulls together all these earlier points, which is the challenge of integrating data sets. In non-precision medicine, we have the virtue of relying on trials that all are fairly large clinical trials, low degree of variance because they're large trials. And ideally, we can synthesize or pool fairly similar kinds of data sets into a meta-analysis to form a judgment about the clinical utility of a treatment. That is not a luxury that we necessarily have available to us in the context of precision medicine, where we may be pooling small trials and trying to aggregate or interpret them in light of outcomes in preclinical research, other studies, trials testing similar but non-identical drugs, or trials testing identical drugs with different diagnostic techniques to match patients. And so one of the challenges here is that we have to create or develop different ways to aggregate information from disparate sources, something that's really less familiar to the practice of non-precision medicine. I just want to mention two other really quick points that I think in precision medicine are creating pressures on preclinical research. There's this category of clinical trials, these platform trials, where we have these arms that are being added or closed. The basic idea of the platform trial is to create this infrastructure that is constantly accruing new hypotheses to test and closing them out, depending on whether they show activity or they show futility. Well, if you invest in creating a large infrastructure like a platform study that has to be running continuously, you're always going to need to be fueling that platform trial with new hypotheses to test. And I think that also creates some pressure on making sure that we have interesting hypotheses to feed into those platform studies, hypotheses that have an adequate risk-benefit balance for the patients who are going to be entering into them. And of course, we also talked a little bit here at this meeting about the avatar or about the patient-derived xenografts. This is another area where there's a tighter coupling of preclinical evidence and experimentation with clinical judgment. I think in medicine, we have this fiction that we've got preclinical evidence, and we have this really good filter between spurious preclinical findings and clinical practice, and that's clinical trials. So, drugs, we get interesting activity in a preclinical study. They get put into phase 1, phase 2, phase 3 studies. That filters out spurious findings from preclinical. Patients are protected from spurious findings in preclinical. And that really has always been a fiction, but I think it's especially a fiction in the context of precision medicine. A non-precision example is the example of a drug called bixaritine, a drug that showed activity the drug that is licensed for the treatment of T-cell lymphoma, it showed activity in a Science article against Alzheimer's disease. On that basis, there was some discussion about trying to actually use this drug off-label in Alzheimer's patients. Just an interesting footnote, to my knowledge, this study actually was not able to be reproduced. But this is also a recurrent theme in the context of precision medicine. Here's a publication looking at pediatric solid tumors where the researchers were looking to identify clinically actionable mutations in a sample of about 100 pediatric solid tumors. And they classified the clinically actionable mutations on the level of evidence. What you see here is that so only 30 out of 100 tumors showed clinically actionable mutations, but most of the clinically actionable mutations were deemed actionable on the basis of preclinical or even level 5, which was expert opinion. I'm not sure exactly what expert opinion means, but anyway, without preclinical. Okay. And it's also very easy to go to the literature and to find lots of case reports, almost all of them positive, by the way, that show that a drug can be used off-label successfully to treat an individual patient on the basis of some kind of preclinical study. So I think in precision medicine we have this coupling, this sort of, you don't necessarily have to go through clinical trials to get to clinical practice, and I think this creates some concerns, or at least it creates some real pressures on getting the preclinical evidence exactly right. Okay, the second part, I want to go through really, really quickly some factors that I think challenge our ability to make clinically generalizable inferences from preclinical evidence. If you think about any kind of preclinical study or any kind of research program, you can divide it into sort of 3 cycles, and there were threats that are introduced at each of the different steps within this cycle. With respect to design, Uli already talked a lot about some of the various threats. There are any number of different variables that can confound our ability to measure a real treatment effect. Internal validity refers to our ability to measure an actual relationship, cause and effect relationship between a drug and some kind of a disease response. In cancer, it's extremely rare to find implementation of measures to neutralize those threats to internal validity. So for example, in my own systematic review of preclinical studies for sorafenib, 0% of studies in at least my review used any kind of outcome assessment blinding. Construct validity, at least in my definition, refers to the concordance between the experimental system and the clinical scenarios they're intended to simulate. Again, in cancer, we've heard about this already, high reliance on juvenile mice, female mice. Most mice are immunosuppressed in the context of cancer. And external validity refers to the robustness or the ability of a cause and effect relationship to withstand perturbations in the experimental system. And again, in my own systematic review of sorafenib, we see that most malignancies that were shown to respond to sorafenib were only validated in a single system. Very rare do you find 4 or 5 different systems used to validate a drug's activity in cancer against a malignancy. Reporting has already been talked about a bit as well. There's different kinds of reporting threats. I'm just going to mention really quickly the problem of non-publication. There's a tendency to publish positive studies, to not publish negative. This is a real problem in the context of precision medicine, I fear. In our own study of sorafenib, we found that if you statistically correct for non-published preclinical studies of sorafenib, the effect size, the mean effect size, drops by about 37%. By the way, if you're interested in this work, it was published in Cancer Research, I think now 2 years ago, and it was done by my master's student, James Mattina. I want to spend a little bit of time talking about the challenge of uptake, something that people don't really talk a lot about, but I think really matters a lot in this context. So obviously, if you do an experiment, the experiment is only valid insofar as you have interpreted or made accurate clinical inferences about what that experiment shows. So an experiment can be completely fine experiment, but if you're interpreting it wrong, then there's a problem. And I think uptake is the missing link in the discussion we have about reproducibility. My own team has tried to actually systematically study the quality of the inferences that we make in the context of preclinical research. Probably people here are aware of the Reproducibility Project Cancer Biology. This is an initiative to take several high-profile cancer studies that were published in high-impact journals and to reproduce them in independent laboratories. What we did is we piggybacked on that reproducibility project and asked a series of experts in cancer biology who had expertise in the individual studies in the reproducibility project to predict whether or not the studies would reproduce using two different measures, whether the studies would reproduce the statistical significance as well as the effect size. So our study looked at 138 different individuals who are identified as having expertise in the areas of the 6 preclinical studies that we studied. We also did the study in trainees. And so what we asked them is to provide a number between 0 and 100% that the studies would reproduce by those two criteria. 66% means that they were betting 2-to-1 odds of the study reproducing. And what you see, these are histograms of people's predictions, and what you see is that people were fairly optimistic. They were closer to 100% that studies would reproduce their statistical significance. They were a little bit more pessimistic, but there was still quite a bit of optimism, a lot of uncertainty at 50% with respect to reproducing the effect sizes. If you actually score the accuracy using a technique called the Breyer score, the accuracy of people's predictions, with Breyer scores, the best predictors will have a score of 0, and the worst predictors will have a score of 1. So you see with respect to, and this line right here, this is a benchmark that represents what you would score if you just guessed 50% every time you were asked a question. What you can see is that for significance, most people did a lot worse than just guessing 50%. And a lot of people did worse with effect sizes, not quite as many people. But if you combine these, generally speaking, expert scientists perform worse than just guessing 50% about whether studies will reproduce, at least in the 6 studies that we looked at using the techniques that we looked at. Now, discouragingly, we wanted to look at whether there were properties of scientists that were better at predicting whether studies would reproduce or not. Unfortunately, what we found was that people who claimed to have greater confidence in their predictions did not necessarily do any better than people who had low confidence in their predictions. If anything, highly confident scientists did worse in their predictions. People who claimed to have more expertise in the individual studies that we were asking them about didn't do any better than people who claimed to have less expertise. The one variable that's a little bit suggestive, it's a small study, so it needs to be reproduced, is the H-index. People who had higher H-indices seem to be a little bit more jaundiced with respect to the likelihood that studies would reproduce. Consequently, people who had higher H-indices tended to actually have more accurate predictions. H-index, for those of you who don't know it, is a measure of scientists' productivity. And that's published in PLOS Biology in case you're interested, and it was led by my postdoc, Danny Benjamin. Let me now try to sort of pull this together and offer a couple recommendations for how I think we might want to try to approach preclinical research in the era of precision medicine. So my first recommendation is, there's all sorts of different ways of doing preclinical research, and there are good reasons to have fairly permissive standards for preclinical research in an exploratory context. But the minute we're talking about trying to use preclinical findings to inform clinical decision-making, I think we really have a strong demand for what I'm calling clinical-grade preclinical evidence. By that, I mean studies ought to have a more kind of confirmatory structure. Uli and I actually wrote a paper a few years ago about describing exactly what we mean by exploratory preclinical studies in contrast, confirmatory in contrast with exploratory. But in essence, what we mean is that these studies ought to be powered a priori. They ought to implement measures like blinding and randomization. The hypotheses ought to be pre-specified, and there ought to be some mechanism to prospectively register these in a database so that we can protect against publication bias. My second recommendation is that we really need to do something to address the quality of the reporting, and I mean this both in the context of preclinical as well as in clinical. In my own research, we've looked at the fraction of trials for drugs that don't get FDA approval, the fraction of those trials that get published. And what we find is that only 37% of trials of drugs that don't get FDA approval get published within 5 years of their completion of enrollment, in contrast with 75% for drugs that are licensed. Now again, if you go back to what I was saying earlier about the need to integrate different kinds of data sets to form inferences about the utility of a drug in a patient population, the fact that we can't rely on large populations if we have these finer and finer grained strata of patients, we're losing a huge amount of information from these unsuccessful drug development trajectories. To me, it's unconscionable that we tolerate this degree of non-publication in the context of drug development. Also an issue, obviously, with preclinical, I've talked about that. Obviously, we are dealing with highly biased data sets if we're trying to go to the literature and to look at the properties of a drug against a particular patient stratum using animals. And my third recommendation is getting back to that issue of uptake. So I think we need to think about ways of improving our ability to interpret and make inferences using preclinical evidence. This is a small study that looked at the ability of garden variety oncologists to make recommendations on clinical actionability in precision medicine context that were concordant with expert judgments at Memorial Sloan Kettering. And essentially what you see is a finding that physicians may have a less stringent definition of actionability than expert curators. Well, that's a concern given that by definition experts are rare. It's mostly going to be those physicians that are making the bedside decisions. There's a large literature on how to improve the ability of people to make predictions, to make inferences, et cetera. And I'm just going to say really quickly that one of the themes that comes up in this literature is that the best kinds of decision and prediction contexts are contexts in which the human is combined with the machine. And I think we need to really think increasingly about ways to improve the human interface with the machine in order to make good clinical judgments in the context of precision medicine. And they're essentially in that sort of human-machine interface, there are two different ways we can intervene to improve the accuracy and the quality of the judgments that are made with respect to clinical utility. The first is you can improve the decision maker. You can train the decision maker on the content of the preclinical research. You can also give them cognitive training. There are certain kinds of heuristics or biases that people can be trained to avoid or to utilize to make better decisions. A third point is that you can give them feedback. Weather forecasters are extremely good at predicting the weather, contrary to popular belief. And part of the reason why they're so effective is because they get feedback every day on how accurate their forecasts were. We don't necessarily have that system set up in medicine. If we did have that system in medicine, we would have a mechanism where people could improve their ability to interpret evidence and make clinical judgments in light of it. The other point where you can intervene, and this is my second to last slide, I have 3 seconds left, is improving the machine. And here I think, thinking about ways that to facilitate an interface of a human being with a data set. The data set or the machine needs to have better evidence put into it. It also needs to have more evidence that's noisy evidence culled from it or removed, evidence that is distracting. And I think getting back to one of those algorithms I mentioned from the University of Toronto, where they excluded preclinical, at this point that actually might be a prudent policy given the amount of noisiness in the preclinical evidence that we have right now in cancer. So I'm just going to close by thanking the two sources of funding that I've had to think about these issues in precision medicine. Thank you.